All right. Well, hello, everybody. And we are live. We have with us Dr. Lissel Shoda. She's the Associate Vice President and Director of Immunology at Simulations Plus, Inc. And she's going to talk about yo-yo dieting predicted to contribute to fibrosis score reductions in untreated placebo cohorts. And I will share again, we're having a little bit of issues with the sharing. So there we go. I hope you can see it. So
I can. Brilliant. Thank you so much. Good morning, good afternoon, good evening to the audience. Thank you so much for joining and for allowing me to share this work. Next, please. Just this work is the hypothesis exploration, but definitely should not be considered medical advice. And so that's just our disclaimer to that effect. Next, please. Okay, so the disease area we're going to talk about today is non-alcoholic fatty liver disease or non-alcoholic steatohepatitis, NAFLD and NASH. And NAFLD-NASH represents a spectrum of chronic liver disease. As you can see in this top right figure, these are histology from liver, and on the left you can see what a normal healthy liver looks like, and then there's a spectrum where that includes steatosis or fatty liver, and then also includes steatohepatitis, so liver disease in which you also, in addition to the fatty liver, have inflammation and fibrosis, and that fibrosis can really become quite extensive, leading to compromised liver function. So NAFLD is something that is widespread and growing. So not to make a pun, but there you go. It is, accounts about a quarter of the adult population in the world, as you can see from this figure on the bottom right. And there are no approved drug treatments. So more typically, it's a suggestion for weight loss by a diet or changes in activity. But that also means that there's really a big opportunity here to improve health for a lot of people if treatments can be developed. So at this point, I believe a poll will launch to just query the audience as to the interest in treatment of NAFLD-NASH in your organization.
people should be able to see the poll right now. We will give them a couple of seconds to answer. And then close it. So we have a very even split at 50-50. Two companies are engaged in development of drugs for treatment of NAFLD or NASH, and 50% are not.
Okay. All right. Let's keep moving then. Next, please. Okay. So there are, as we just saw in the poll, many organizations that are interested in the treatment of NAFLD-NASH. I wanted to take just a moment to talk about histological endpoints because this will be relevant for later in the talk. So liver biopsy is the definitive technique for diagnosis and classification of NAFLD-NASH. There are a few different ways in which that's assessed. One is the NAFLD Activity Score, or NAS, which is a summary score that reflects steatosis, hepatocellular ballooning, and lobular inflammation. Another evaluation that's done with these liver biopsy is to stage the fibrosis. And so there's a scoring system, and you can see sort of example of how that maps against these different stages on the right, where we're not only characterizing the quantity of fibrosis, but also where it's located in the hepatic acinus. And so, in stage 1, fibrosis tends to be more central lobular. Stage 2, you can get it in both central lobular and periportal regions. Stage 3 is bridging, and stage 4 is cirrhosis, essentially. And so you can see that there's both the qualitative and quantitative differences as one progresses in disease. Next, please. Okay, so clinical trials, we just said that there are many organizations that are interested in developing treatments. One of the things that we've noticed in looking at the clinical trial data are that there can be a fair number, 10 to 20% of the patients in the placebo group that demonstrate improvement on the fibrosis endpoints. And so on the left, in both the left and right figures, which represent two different clinical trials, the placebo group is indicated in the open bars, and you can see that that varies between, say, 10 and 20%. And we could argue about whether that is real. Perhaps there's some differences with respect to histological assessment. But we really had this question about what is going on with those placebo patients. There's also sort of this general historical thought that fibrosis tends to be more unidirectional in its progression. So it's sort of a surprising result. Next, please. And so that really led us to sort of wonder what might be going on with respect to these placebo-treated patients, and why might they, if it's real, right, why might they be seeing improvement in the fibrosis endpoints? Next, please. So it turns out that we have a QSP model of NAFLD, and it's called NAFLD-SIM. And we really think that this is a fantastic tool to be able to evaluate this particular question. Next, please. And actually, Amparo, would you mind just go ahead and building this one out? So it should be 4 clicks. NAFLD-SIM includes elements of steatosis. So in the diagram, that's indicated in sort of the open boxes, as well as lipotoxicity in the red boxes. We have elements of inflammation in blue and elements of fibrosis in green. And this is all built on publicly available literature to define the different components, quantitative relationships among them. And we have developed simulated patients that represent variation in many of those parameters and reflect the variability that you see in the patient population. And these patients in the simulated population in general have been compared against many publicly available data sets in terms of individual component parts and sort of where they're sitting. So for example, the levels of inflammation, macrophage numbers, the TNF alpha levels or TGF beta levels, et cetera. But also looking at, and this has been used to evaluate treatments, so to predict efficacy for various treatments. And there are many examples of that have been presented in posters. But we're talking not so much about treatments, we're talking about the placebo group. So next, please. We are building on a little bit of work that's been done in this space with NAFLD-SIM. So this is a screenshot of a poster presented at ACOP in 2019, and really looking at developing patients and looking at disease progression. And as part of that, we can see changes in those histological endpoints like NAS and fibrosis stage. Next, please. And then there's also been work to look at, in the context of NAFLD-SIM, what happens with weight loss and what kind of patient variability do we see in the various endpoints. And so next, please. We really thought, okay, so we can build, stand on the shoulders of giants and build on this to really start to query, hey, what might be going on in that placebo group that would account the 10 to 20% that have a improvement in fibrosis. And So at this point, next please, what I'd like to introduce is this concept of yo-yo dieting. So yo-yo dieting is a terminology that was coined to um, characterize the phenomena of intentional weight loss followed by unintentional weight regain. And so, individuals may be advised by their physician to, lose a little bit of weight. And so, people try and do that through diet or activity, and then, but it's not an easy thing to do, and often there's weight regain. And it's been actually associated with sort of negative connotations in the historical literature. Although recently there are a few efforts that have sort of suggested, well, maybe it's not as clear that it's a negative consequence. And so our hypothesis was, okay, in a clinical trial, the participation where you are going in, getting evaluated on a regular basis, maybe that alters people's behavior a little bit so that you have some unintentional weight loss. And then maybe you go to your regular visit and you're like, okay, great, everything's going well, and maybe you relax a little bit, and there is a little bit of weight gain. Now, we don't think that it would be really a extensive weight loss and weight gain, because if it was really simple, like that, then it might be picked up, right, by the physicians, by the researchers. So yo-yo dieting, this idea is really to sort of look at subtle changes that might manifest as more profound changes in our endpoints. Next, please. Okay, so how did we tackle this? We identified a set of simulated patients, sim cohorts. This was 90 different individuals with the characteristics that you see in the table. One thing I will point out is that, in that fibrosis spectrum, that all of these patients were either F2 or F3, with more F3 patients. So we're looking at patients that are a little bit farther down the spectrum of fibrosis. These are also the patients that tend to be targeted for in clinical trials, although sometimes there are F1 patients as well. Next, please. And then with respect to how are we going to do yo-yo dieting? So the plan was to do a very modest 1% weight loss over 4 weeks, and then follow that by a 1% weight gain over the next 4 weeks, and to repeat that cycle over a year period. And so again, really modest changes, right? But and see, how does that manifest? Okay, let's find out. Next, please. All right, so with successive cycles of really mild weight loss and weight gain, in these 90 simulated subjects, we look at these clinical endpoints, and actually, because it's a model and simulations, we can look at this over time. And so this is what we're looking at in the right table. We have time on so 13 weeks, 26, 39, 52, and the percentage of the simulated patients that have a reduction in their fibrosis stage of at least 1 point. So it could be more than one. You could go from an F3 to an F1, or you could be going from an F3 to an F2. But that's what that's representing in that column. And what we can see is that as early as 13 weeks, there are 6% of individuals in this simulated cohort that have a reduction in fibrosis stage. And if you follow that out at 52 weeks, it's a persistent response where we now see about 10% of individuals that have undergone a reduction in fibrosis stage. So it's quite similar to what we've seen in the clinical reports. By contrast, we're not seeing anything in the placebo cohort with respect to NAS. And you'll recall NAS is really sort of looking at things like steatosis, inflammation, which, might fluctuate a little bit more. So, there's this interesting dynamic where we're seeing, an effect on the fibrotic endpoints, but not so much in the inflammatory slash steatotic endpoints. Okay, so that is sort of the big picture finding. I really want to dive into the details with the group and sort of show at least one level deeper what's happening. But let's pause for another poll question.
And you will be seeing that in your screen right about now. Let's give people a couple of seconds to answer to that. And the question is predicting drug and or placebo responses in NAFLD NASH patients of interest to you or your company? Because I'm sure we can help you out. So we'll end the poll. And we have a very overwhelming majority of 80% saying yes. So if you're not in contact and in collaboration with us yet, please do go to our website and contact us. We'll be happy to help.
Okay, next, please. So in analyzing, simulation results, one of the things that you typically want to make sure you do is, are you doing what you think you're doing, right? And so, we're really just in this particular first slide, just illustrating that the weight changes that were intended are in fact manifesting in the cohort in the way that we had designed them to do. And so on the left figure, we're looking at the percentage body weight change over 52 weeks. And you can see there's sort of in that first 4 weeks of fluctuation down that then returns over the next 4 weeks, and that cycling continues by design, right? And if we look at on the right side, the body weight across the cohort, we can see that, yeah, those fluctuations are evident in the cohort, but you can pretty well imagine that that's not something that can be clinically picked up, right? It's very subtle. Okay, so we're doing what we think we want to do. Next, please. Let's look at some of those endpoints that really didn't change too much at the high level, or at least one level deeper for those endpoints. So on the top 2 figures, we're looking at liver fat change as a percentage or an absolute value. And on the bottom, we're looking at plasma ALT, again, as a relative difference or as absolute values. And liver fat being related to steatosis, plasma ALT being sort of a function of hepatocyte death, and so therefore probably related to hepatocellular ballooning. And what we can see is that there are oscillations seen in both of these, and those oscillations really seem to follow the weight loss, weight gain paradigm. And so they're sort of similar. We're going back and forth and following things with very minor changes in the absolute values. Now, one thing that's kind of interesting about this is that, so the fact that they're, following the weight change oscillations, the very minor differences between when you started and at 52 weeks. So, wouldn't necessarily expect to see differences. But you can see, because there are oscillations, depending on the endpoint, depending on the particular individual and their makeup, you might actually see, some changes depending on when you sample or the patient, the particulars of the patient. Next, please. Now we'll contrast that with liver collagen. So liver collagen, as most of you will know, is a constituent portion of the extracellular matrix. It's really part of what makes up that fibrotic tissue. And so if we look at liver collagen, the dynamics are completely different than what we looked at on the previous 2 slides. So there is a delayed response. That response is there is a decrement in the liver collagen, and that is persistent over time. So this really demonstrates that, and I think, those of you from the modeling world really appreciate this, and many others as well. Dynamics matter. Some things happen on very short time frames, and other things happen over much longer time frames. But it's really hard to appreciate those differences in our mental models of what is happening. And so what this is, illustrating in sort of stark form is that what's happening with the fibroblasts, the hepatic stellate cells, and the laying down or degradation of extracellular matrix is really happening on a more chronic timescale than what we're seeing in the steatosis and in the inflammation. The other aspect of this is that, again, the fibrosis scores not only reflect amount of collagen, but also localization. And so that may be, partially what's at play here. There might be a difference between, if you have a little bit of bridging necrosis versus not having bridging necrosis. And again, this is dependent on biopsies, so we know that these are relatively small samples. Next, please. Okay, and so just to pull this back, to that whole dynamics and how it compares then, what we're looking at is now on the time scale basis and a little bit more granularity, the NAS on the left and the fibrosis score on the right, or the percentage of patients with a reduction in either NAS or fibrosis score. So this sort of, again, highlights the point that depending on when you look, you might see a difference in NAS. But the dynamics really follow the weight loss and weight gain, whereas with the fibrosis, we have more of a persistent response. The oscillatory behavior is clearly on a very different time frame, and something that is more likely to be picked up in a more robust way over that year-long period. I will comment that this is clearly an artificial scenario. We're imposing 1% exactly, right? Weight loss and weight gain in a very systematic way. So you can imagine that different weight loss, yo-yo cycling paradigms could give you somewhat different results. Okay, next, please. So, in summary, we were able to use NAFLD-SIM to explore the hypothesis that yo-yo dieting might be a difficult to detect, but meaningful contributor to endpoints as seen in the placebo cohorts. It really has to do with the slow rates of change in collagen versus some of the other measures that can change more dynamically with the weight loss and weight gain, and that really reflects the ability to see these. Next, please. So I'm just the voice of this. This is really work that was inspired by Scott Seiler, who is our primary architect of NAFLD-SIM and really our leader in this particular area, with big contributions from Christina Batista, Grant Genero, and a number of other people at DILIsym who contribute to the development. This poster was presented at AASLD last year, the virtual meeting there. And I would be happy to take any questions. I think there's one last slide, but that's just the question slide.
Thank you very much, Liesl, for a wonderful presentation. We do have two questions. Actually, the first one is there a detailed biochemical explanation to how exactly the yo-yo dieting influences hepatic collagen?
Yes, there is. It might be a little tricky to answer that in the time we have available, but essentially what it has to do with is you remember the diagram of NAFLD-SIM as a whole. So all of those parts are interconnected. And so if you alter things on the, with respect to the steatosis and the fatty hepatocytes and the hepatocyte death, that will in turn affect the inflammation, which in turn will affect the activity of the fibroblasts. And so you get changes in not just the activity, but also in multiple mediators that regulate that activity. And then that sort of affects the rates of collagen, not just collagen synthesis and degradation. So, you know, we have the fibroblasts making collagen, things like MMPs degrading it, or neutrophil elastase degrading it, the TIMPs that are modulating the MMP. And so there are sort of all those interactions that are affecting the hepatic collagen. And then in terms of like how this manifests in time, you know, that's really informed by, and I didn't speak to this, but there is literature out there that have used sort of weight loss on a much more, you know, measurable and larger scale, and looked at, you know, the influence of weight loss or weight gain on fibrosis, and so, or, you know, NAFLD in general. So there are sort of constraints to help us to understand how all those detailed biochemical interactions really manifest in you know, the timing with which you can observe differences.
And then we have another one, and we have the panel at the end of all the poster sessions that come in next, so we can talk more about and ask more questions to all of our speakers, which will be panelists. The other one was, can you comment on any key clinical trials that have been impacted by this observation?
So I'm going to interpret that as the observation that there is a relatively large placebo response. Mm-hmm. And the answer is yes. So one of the clinical trials that I showed a figure of early in the presentation were the results from the Centaur trial. And this was a clinical trial evaluating Senecrevirac, which is a CCR2, CCR5 antagonist. And the expectation was that it was going to influence both inflammation and fibrosis. And it was, in terms of the, and actually they had initially sort of prioritized the inflammation endpoints, but it turned out their initial sort of interim analysis suggested potential fibrotic improvement. Very exciting stuff. And then when they, you know, sort of followed this and got the full analysis, they found that the placebo response was significant enough that if there was a Senecrevirac response, it was difficult to tease out from the placebo. And so definitely, you know, there can be instances where if we understood this better, if we could figure out a way to sort of minimize that placebo response, it would be easier to understand, you know, the true therapeutic efficacy of a particular compound.
Very good. And we're almost at the time. Thank you so very much. And we'll keep on talking at the panel. For everybody else that is attending, please go back to the lobby to log on to the next talk. Thank you.
Thank you.